Hello and welcome back to my channel. And today we're going to start the PT365 for the one and only subject Science and Technology. So, as you all know, that I am kind of lethargic reading it alone on my own. So, I'll just have you guys as my study partner for reading this very document. Without any further ado, let's get started. So, the first topic is about biotechnology, genes and related concepts. Genes are segments of deoxyribonucleic acid. DNA is an important nucleic acid found in human cells along with the ribonucleic acid RNA. So DNA is basically double stranded. So it is kind of in a helical structure, somewhat like this. It's not very good at drawing, but this works, right? So chromosomes are thread-like structures made of protein and a single molecule of DNA. So I'm kind of bored by this very color. I'll just change it. So this looks good. Kind of in with darker ranges. Yeah, this one's fine. Okay. Yeah. Anyways, let's go further. Genome is the entire set of DNA instructions found in a cell. Comparison between DNA and RNA. So both of them has been compared along several parameters, which are the structure, nitrogen species, and functions. So DNA is double-stranded helix, as I showed you earlier. RNA is single-stranded. DNA comprises of the nitrogen species as AGCT, adenine, thymine, cytosine, guanine. So you might have read this in your uh, class 2 chemistry classes. RNA has uracil in place of thymine, so it is AGCU. DNA stores genetic information for inheritance. RNA regulates gene expression and plays key role in protein synthesis. So the central dogma of genetics is DNA to RNA to protein. So this is the central dogma. Genome editing, also known as gene editing, enables scientists to change on organisms' DNA sequence, leading to changes in physical traits like eye color and disease risk. Uses size-directed nuclease to make changes that may either be a small deletion, a substitution, or the addition of several nucleotides. So these are just trying to add or delete the sequences which we want. Histine refers to the practice of cleaving DNA strands to affect the subsequent genome editing. So site-directed nuclease. This is the one which we use to cleave the DNA strands. The key gene editing technologies are CRISPR-Cas9, the most upfront technology right now. CRISPR-Cas9, which is the full form is clustered regularly interspaced short palindromic repeats. CRISPR-Cas9. So palindrome is basically uh, it's a sequence of numbers which we term as palindromic sequences. So for example, a number that remains the same when its digits are reversed. Let's take 1331. Even if you reverse this, it would be 1331. So the reverse digits as well as the original number, both of them are same. This is what is a palindrome. So if even if you read it backwards or forwards, it still gives the same answer. This is what we call as the palindrome. Anyways, the other editing techniques are zinc finger nuclease, ZFNS, transcription activator like effector nuclease talents. Just remember the short form talents. Genome editing is different from genetic engineering, genetic modification, because to modify is like you are actually adding something or adding a foreign material. So genome editing is different from genetic engineering. in the sense that genome editing does not involve the introduction of foreign genetic material called transgene, and the latter does. So, in genetic engineering, there is actually an introduction of the foreign genetic material called transgene. However, in genome editing, there is no addition of any kind of foreign material. There is no addition of transgene. CRISPR-Cas9, as we have read earlier, clustered, regularly interspaced palindromic repeats. It changes genetic code or edits DNA at particular locations. So in this, the Cas9 protein is actually the molecular scissors. We have actually had a previous year question regarding this. It changes genetic code or edits DNA at particular locations. Works as cut and paste mechanism on DNA strands. The genetic codes that need to be changed are identified. Cas9 protein is used as a pair of molecular scissors to cut off apart from strand, allowing modifications to the genome. Genome sequencing determines the exact order of base pairs in an individual's DNA strand. So there was also a question about DNA barcoding. So where you actually, what is a barcode? Barcode is basically trying to synthesize or consolidate certain information in a very small rectangular code, right? So similarly, genome sequencing is like we're trying to keep the base pairs in order. We're trying to arrange them in certain order in order to use them or extract them for different purposes, be it scientific or be it other purposes related to health. Applications of genome sequencing, predictive diagnostics, and personalized healthcare. So, just it is the customization of healthcare. This is why we use genome sequencing, predictive diagnostics, and personalized healthcare enables treatments for genetic diseases. Paternity testing confirms biological relationships. Agricultural identify traits for higher yield, disease resistance, and climate resilience. The next article is about the Nobel Prize in Medicine for 2024 gene regulation. Nobel Prize in Physiology or Medicine has been awarded to Victor Ambros and Gary Rafkan for the discovery of microRNA and its role in post-transcriptional gene regulation. So it is basically into news because for the Nobel Prize given to the two people in the field of physiology or medicine, which are Victor Ambros and Gary Rafkin, for discovery of microRNA, and it has a prominent role in post-transcriptional gene regulation. So after the gene is transcripted, this is where micro DNA comes into play. Discovery of Victor Ambros and Gary Rafkin. So, what did they actually discover? In 1993, they discovered microRNA and its role in gene regulation after transcription. Till 1993, it was believed that gene regulation is limited to specialized proteins called transcription factors, which bind to specific regions of DNA and determine which messenger ribonucleic acid is produced. So, till 1999, it was believed that the gene regulation was only limited to some specialized or specific proteins called the transcription factors, which would then bind to specific regions in the DNA and which would later determine to which mRNA will be produced. They discovered the role of discovered the role of microRNA by investigating mutant Cyanohabditis elegans nematode. So C. elegans nematodes. About microRNA, the small non-coding RNA that helps cell regulate gene. So the microRNA controls gene expression by binding with mRNA and preventing them from being translated into proteins or by degrading or destroying mRNA altogether. So this is a very important role of microRNA, and this is why this discovery is of importance to us. So microRNA is a non-coding RNA and it helps in the gene it helps in regulating the gene expression. So what it does is it controls the gene expression by sticking or binding with micro by sticking or binding with mRNA. So micro RNA actually sticks with mRNA and prevents it from being translated into proteins or degrade or destroy mRNA altogether. So it basically prevents the synthesis of protein. So in the central dogma of molecular biology, we read that DNA then leads to transcription, leads to RNA, which by translation leads to proteins. So what the microRNA does is microRNA actually sticks to mRNA and disrupts this process. So it basically disrupts this process and now protein is not formed. So the gene is not expressed. Proteins in the nucleus regulate RNA transcription and splicing with microRNAs control the translation and degradation of messenger RNA in the cytoplasm. So proteins in the nucleus regulate RNA transcription and splicing, while microRNA controls the translation and degradation of mRNA in the cytoplasm. About gene regulation refers to the process that controls the timing, location, and amount in which genes out of many genes in a genome are expressed. Gene expression. Human organs and tissues consist of different cell types. The chromosomes in a cell contains the same set of genes with the same set of instructions. However, due to gene regulation, these different cells like muscle cells, nerve cells, etc. express unique set of proteins, enabling them to perform their specialized functions. So this is the DNA transcription for mRNA, then translation, then protein. So till 1993, we read that it was believed that gene regulation is limited to specialized proteins called transcription factors, which bind to specialized areas or specific regions in DNA and determine which messenger ribonucleic acid is produced. So basically, what it means is there were only certain proteins which were responsible for the transcription, and they would later determine where to bind in the DNA and uh, which messenger RNA should be produced. So, what microRNA does is it sticks to this part of mRNA and prevents the gene from being translated. So, this won't cross this process. Since the gene won't cross this process, the protein won't be synthesized, and this can be done by preventing them from translated into proteins or by degrading or destroying the mRNA altogether. It can also degrade or destroy the mRNA, the microRNA can. So human genome contains 20,000 genes. Significance or applications of discovery. Understanding cellular development, example self-renewal and differentiation of the stem cells. Immune response, regulate innate and adaptive immune responses. Oncogenesis to avoid healthy cells conversion to cancer cells. So to prevent the expression of some onco cells, we could use the microRNA. And this is diagnostics example cancer. So this is very pivotal in cancer diagnostics. Related concepts, reverse transcriptase. So just found that when infected by a virus, bacteria, Klebsiella pneumoniae use RNA to bind reverse transcriptase. About reverse transcriptase, they are RNA dependent DNA polymerase, a group of enzymes that play a unique role in the flow of genetic information. These enzymes enable the reverse transcription reaction. Reverse transcription is the synthesis of DNA from an RNA template. So basically, it is this process from RNA to DNA. I'll just show you in a different color. From RNA to DNA, this reverse transcriptase. So, RNA-dependent DNA polymerases, a group of enzymes that play a unique role in the flow of genetic information. Genetically modified crops. The Supreme Court pronounced a split verdict on the validity of the Union government's decision to grant approval for the environmental release of genetically modified mustard crops in 2022. So, what are these genetically modified crop? TMH 11, Thara mustard 11. Developed by Center for Genetic Manipulation of Crop Plants, Delhi University, GM mustard has not been released for commercial cultivation yet. It is India's first indigenously developed GM food crop. DMH11 is a result of a crossing between two mustard varieties, Varuna and East European early Hira. So Varuna and Hira are the two varieties, and a mixture of this or the crossing between these two led to the formation of DMH11. This cross has been done after introducing the Barnase and bar star gene from the soil bacterium Bacillus amyloliquefaciens. So the two genes are the Barnase and bar star, and these are extracted from the soil bacterium Bacillus amyloliquefaciens. Other genetically modified crops in India. BT cotton. This was the first one. Non-food only approved GM crop in 2022 for commercial cultivation. It was introduced to protect against the widespread infestation of bollworms such as pink bollworm (PVW). Then is the BT brinjal. In 2009, BT brinjal was cleared by GEEC for commercial cultivation, but it was put on a moratorium. So it was approved by the GEEC, but it was put on a moratorium later. So moratorium is basically a temporary stopping of an activity. So they're just stopping its activity for a temporary basis by an official announcement, official degree. Benefits of GM methods in agriculture: enhanced yield protection, that is resistance to pests and diseases, reduced usage of pesticides, enhanced nutritional value. Example: golden rice. This is a rice fortification, addition of vitamin A into rice, and the tolerance to drought. So these are the four major benefits of genetically modified crops in agriculture. So first is the enhanced yield production, more yield. Next is the reduced usage of pesticides, low pesticides, enhanced nutritional value, and drought tolerance, drought resistance basically. So what is the regulatory framework of GM crops in India? First is Food Safety and Standards Association Act, Safety and Standards Act 2006 prohibits import, manufacture, use or sale of GM food without FSSAI's approval. Review Committee on Genetic Manipulation (RCGM) under the Department of Biotechnology. This committee monitors various aspects of research and development projects involving GM organisms. State Biotechnology Coordination Committee (SBCC) reviews the safety and control measures in various institutions handling genetically modified organisms (GMOs). District level committee. So there are four-tier framework. First is the FSSAI's approval. You need FSSAI's approval. Then is the Review Committee on Genetic Manipulation, which is under the Department of Biotechnology. There is the State Biotechnology Coordination Committee, and next we have the District Level Committee, which investigates, inspects, and reports to state State Biotechnology Coordination Committee or the GEAC, Genetic Engineering Appraisal Committee, about the compliance. So, Genetic Engineering Appraisal Committee (GEAC) is a statutory committee constituted under the rules for manufacture, use, import, export, and the storage of hazardous microorganisms, genetically engineered organisms, or cells. 1989. The rules are framed under Environmental Protection Act 1986. It falls under the Ministry of Environment, Forest and Climate Change. Its responsibilities are appraisal of proposals relating to the release of genetically engineered organisms and products, appraisal of activities involving large-scale use of hazardous microorganisms, and recombinants in research and industrial production. Next is the heritable human genome editing. South Africa becomes the first country to allow heritable human genome editing (HGE). Unlike somatic cell editing, somatic cells are basically the cells which are different from the germline cells. So the cells apart from germline cells are the somatic cells, which affects only individuals. Unlike somatic cell editing, which affects only individuals, HHGE introduces change in germline cells, sperms, eggs, or embryos, enabling these alterations to be inherited by the offspring. So, the changes in the somatic cells or the editing in the somatic cell could only affect the individual, or could only affect one person. However, if you use HHGE, which is heritable human genome editing, this would change your germline cells. Germline cells are basically the sperms, eggs, or embryos, and this would later lead to alterations in the progeny, or this would be inherited by the offspring. Because obviously, these are changes in the germline cell, and the offspring basically inherits all those traits from the germline cells. Potential application: disease prevention. These includes heritable diseases, uh, Huntington disease, and sickle cell anemia. Assisted reproductive technology can improve technologies such as in vitro fertilization. The concerns: unforeseen consequences. Germline, edits would be heritable, and their effects would be multi-generational. So even a small minor uh, disturbance could lead to affecting um, many generations. Societal impact: creation of designer babies, where traits like intelligence, looks, etc. might be engineered. Next topic is RNA editing. Why news? The first successful clinical demonstration of RNA editing in humans was conducted. Wave Life Sciences has successfully performed RNA editing to treat alpha 1 antitrypsin deficiency (AATD), an inherited disorder. In AATD, levels of protein alpha 1 antitrypsin build up and affect the liver and lungs. So, because of this protein alpha 1, uh, because of AATD, the protein alpha 1 antitrypsin actually builds up, piles up, and then affects the lungs and the liver. About RNA editing, ribonucleic editing, a process that modifies genetic information on RNA sequences through insertion, deletion, or substitution. Scientists use techniques called adenosine deaminase acting on RNA ADAR, adenosine deaminase acting on RNA ADAR with guide RNA gRNA. gRNA are small RNA molecules that direct editing machinery by base pairing with mRNA in specific regions for modification. So they basically are the ones who are trying to direct, or who are trying to show you the exact location where you need to edit. So these direct editing machinery by base pairing with mRNA. Comparison between RNA and gene editing. Uh, Form of change. DNA editing makes permanent change. Okay, whereas while RNA editing makes temporary change. So DNA will lead to permanent change. Thus, RNA editing is safer and flexible compared to DNA editing, which may result in irreversible errors. Allergic and immune reactions. DNA editing has a higher risk of undesirable compare undesirable reactions compared to RNA editing. DNA editing tools use proteins from certain bacteria to perform cutting function, while RNA editing relies on ADAR enzyme already occurring in the human body. So this ADAR enzyme already occurs in the human body. Adenosine deaminase acting on RNA. This is basically present in the human body, and whereas the DNA editing tools like CRISPR-Cas9 use a protein which is not derived in the which does not naturally occur in the human body. For example, the Cas9 protein. They are using different proteins to perform the cutting function. So this might lead to certain immune reactions or allergic reactions. So therefore, the DNA editing has a higher risk of undesirable reactions. Bridge recombinase mechanism (BRM). Scientists discovered a naturally existing DNA editing tool, bridge recombinase mechanism, which utilizes mobile genetic elements or jumping genes. So they have actually discovered a new DNA editing tool, but it was pre-existing. This is not something which is found now. This was pre-existing. It's just that they have discovered now, and this is because of the mobile genetic elements or jumping genes. So the tool name is bridge recombinase mechanism, which utilizes jumping genes. So bridge recombinase mechanism or BRM. BRM is extra DNA ends at the ends of jumping genes gets joined together and converts the DNA double helix structure into single stranded RNA molecule. This bridge RNA molecule can bind to two DNA segments, donor and target, allowing for flexible DNA modifications. So the strands of these jumping genes get joined together and convert the DNA, which is basically in the double helix structure, to a single helix, which is shown over here, and this converts into a single stranded RNA molecule. This bridge RNA molecule can bind to two DNA segments, donor target, allowing for flexible DNA modification. So this would allow in the DNA modification as well. About jumping genes. Jumping genes are DNA sequences that move from one location or the genome to another. These are also known as transposable elements. These small DNA segments contain recombinase enzyme along with extra DNA segments at the ends of the genes that binds and manipulate DNA. They can replicate themselves, insert copies at new locations. Their movement can cause genetic mutations and contribute to genome evolution. So these are very important because they can eventually lead to genetic mutations and later genome evolution. So here's a diagram shown. These are the extra DNA segments at the ends. So one day, one genome. The Department of Biotechnology and Biotechnology Research and Innovation Council (BRIC) launched the One Day, One Genome initiative on first foundation day of BRIC. So Biotechnology Research and Innovation Council. This is the organization. The aim of One Day One Genome is an annotated microbial genome will be publicly released every day to make microbial genomics data more accessible to researchers. So an annotated microbial genome will be publicly released every day. It will highlight the unique bacterial species found in our country. So this would help in the research and development in the field of molecular biology. Molecular biology. About microbial genomics. It is the scientific field that studies the complete genetic material of microorganisms to understand their structure, function, evolution, and interaction with other organisms. So basically, genomics is the studying of genes, and microbials is related to microorganisms. So basically, trying to study the genes of microorganisms, genetic material of microorganisms. Microbes or microorganisms are too small to see with the naked eye, example, bacteria, algae, etc. Significance of studying microbes. This would help in human health, obviously. Understanding the relationship between disease causing bacteria and their genetic makeup. Also, we could use these bacteria in trying to find out the uh, uh, parts of the bacteria which could be useful in DNA editing. We could use proteins from certain bacteria to perform the cutting function for DNA editing. So, next is the advanced biotech applications. Example, biofuel production, biomanufacturing, etc. The environmental sustainability example, Adionella sickensis produces an enzyme that degrades PET. So this will help in uh, reusables. So Adionella sickensis produces an enzyme that degrades PET plastic into reusable monomers. so thereby enhancing the environmental sustainability. This is ideoneuracytokinesis. Human health and disease management. Example, genome sequencing of Mycobacterium tuberculosis. Mycobacterium tuberculosis helps in detecting drug-resistant strains. Agriculture, example, nutrient cycling, nitrogen fixation, maintaining soil fertility, etc. The other developments in the field of biotechnology. First is recombinant proteins. Researchers at the IISc have developed a new process for production of recombinant proteins. So, what are exactly recombinant proteins? Recombinant proteins are proteins that are artificially produced using genetic engineering techniques. These are modified or manipulated proteins encoded by recombinant DNA rDNA. rDNA is an artificially made DNA strand that is formed by the combination of two or more DNA molecules. So, recombinant basically means joining two or more strands of DNA which are basically not found together. So, recombination of two or more strands. So, our DNA is an artificially made DNA that is formed by the combination of two or more DNA molecules. RDNA technology can be used to combine or splice or transfer DNA from different species or to create genes with new functions. Combine or splice or transfer DNA from different species or to create genes with new functions. Production. Recombinant proteins such as monoclonal antibodies are mass produced by growing modified bacteria, viral, or mammalian cells in large bioreactors. Monoclonal antibodies are lab-made proteins mimicking natural antibodies. So these can be like mass produced by growing them in large bioreactors. What are the clear applications of recombinant proteins then? First is the production of biotherapeutics such as insulin. Insulin basically used by the diabetic people, such as insulin growth hormones and monoclonal antibodies for vaccines basically. Next is the development of vector vaccines, considered safer than the traditional vaccines as they do not contain the live pathogen. So they are safer because they do not contain live pathogens, which could, if there was a live pathogen, it could actually infect you further. The next application is in the field of agriculture, used in development of genetically modified crops, increase nutritional value of animal feed, etc. Environmental, used in bioremediation, process of using microorganisms to break down pollutants in the environment. So, in the process of bioremediation. Next is Cipteris, Cipteris oblanceolate, fern with largest genome. So, this has a very peculiar name. Anyway, T. oblanceolate. New research shows that T. oblanceolata, a species of fork fern, has the largest genome. About T. oblanceolata contains 160 billion base pairs, the units that make up a strand of DNA, outstripping the human genome by more than 50 times. Belongs to primordial group of plants that evolved long before dinosaurs set foot on the earth. So even before the dinosaurs were there. This is a species of fern that has the largest genome and which is about 50 times more than that of the human genome. Next article is on the DNA repair. Recently, scientists have discovered a new target for cancer treatment, which is used by cancer cells to regulate DNA repair used during cell division. So, scientists have discovered a new target for cancer treatment, which is used by cancer cells to regulate DNA repair during cell division. So, this is targeting the DNA repair of the cancer cells during cell division. Cancer cells use the enzyme TDP1, triosyl DNA, phosphodiesterase 1 to repair DNA damage, to repair their own DNA damage where the oncogene is there, caused by chemotherapy drugs like camptothecin, leading to treatment resistance. So they basically use the enzyme TDP1 to repair their own DNA damage in response to the chemotherapeutic drugs like camptothecin, leading to treatment resistance. TDP1 is an important enzyme in humans that plays a crucial role in DNA repair by removing damaged DNA bases. About DNA repair. DNA repair is a mechanism of a cell to maintain the integrity of its genetic code. So even DNA knows how to maintain its integrity. It exists in both prokaryotic and eukaryotic organisms. So the TDP1 enzyme is of utmost importance right here because TDP1 is an important enzyme in the humans which basically plays a crucial role in DNA repair by removing the damaged parts of the DNA bases. So the cancer cells are basically using this TDP1 enzyme to repair the DNA damage, to repair their own DNA damage, which was done because of the uh, reactions of the chemotherapeutic drugs like camptothecin. And because of this repair of the DNA, this leads to treatment resistance because it keeps on dividing because of cell division. Next is the plant genome editing tool ISDRA to TNPB. Uh, is ICAR recently developed a miniature plant genome editing tool ISDRA to TNPB. TNPB proteins are considered the evolutionary ancestors of Cas12 nucleases. About genome editing tool ISDRA2 TNPB, derived from bacteria called Deinococcus radiodurans. It can survive extreme uh, environmental conditions, belongs to a family of jumping genes. Jumping genes are the one which we have read earlier. They have more extra DNA material in their ends and they actually stick to the RNA, forming one single pair. Significance is TNPV can target unique regions in the genome that Cas9 and Cas12 cannot. So this is more efficient in targeting the unique genomes than Cas9 and Cas12. Facilitates the creation of fusion proteins, trimeric proteins, created by joining two or more genes that originally coded for separate proteins. Next is the exosomes. An exosome-based gene editing platform, Safe Exo CAS, has been developed by scientists at Columbia University. What are exosomes then? Exosomes are naturally occurring vesicles that have the potential to be manipulated to becoming promising drug delivery vehicles. So these have the potential to become the promising drug delivery vehicles. These are also naturally occurring. Exosomes are membrane enclosed vesicles actively released into the extracellular space. So the next article is on gene therapy. So gene therapy is a technique that uses genes to treat, prevent, or cure a disease or medical disorder. In most gene therapy, a normal gene is inserted into the genome to supplement an abnormal disease causing gene and restore the target cell to a normal state. So, in most of the gene therapies, we usually follow the ROM that the normal gene is inserted into the gene to supplement or augment the abnormal disease causing gene so that we could restore the target cell to the normal state. So, we're just trying to insert a gene to the supplement the abnormal disease causing gene. Gene therapy uses products or methods such as plasmid DNA, genetically engineered circular DNA molecules, etc. Types of gene therapy. First is the germline gene therapy, and the other is the somatic cell gene therapy. In the germline gene therapy, the germline cells, egg or sperm, are modified by the introduction of functional genes. In somatic cell gene therapy, in this, therapeutic genes are transferred to patient somatic cells other than the germline cells. So this is the working of the gene therapy. Cell with non-functioning or this is a cell displayed which has a non-functioning gene. And now we have added DNA containing a functional version of the lost gene in order to augment this and return it back to the normal state. So cell functioning now normally. So what are the applications? Applications are that both inherited genetic diseases, example sickle cell anemia, sickle cell diseases, hemophilia A, and acquired disorders, example leukemia, could be treated. So first article is on the chimeric antigen receptor CAR T cell therapy. India's first homegrown gene therapy, next CAR-19 CAR T cell therapy for cancer, has been launched by the President of India. Next CAR-19 CAR T cell therapy has been developed by IIT Bombay and others. About chimeric antigen receptor CAR T cell therapy. It modifies immune cells, specifically T cells, by turning them into potent cancer fighters known as CAR T cells. So, they are basically modifying the immune cells, which are basically includes the T cells, and turning them into potent cancer fighters. T cells are special cells, type of white blood cells or WBCs, whose primary function is cytotoxic, meaning killing other cells. So this is the primary function of the T cells, and we are basically targeting these T cells in order to turn them into potent cancer fighters, thereby the name CAR T cell. T cells are taken from patient's blood and are changed in the lab by adding a gene for a man-made receptor called CAR. So T cells are extracted from a patient's blood and then they are modified in the lab by addition of a gene for a man-made receptor called CAR. CARs are proteins that assist the T cells to recognize and attach to specific proteins present in the cancer cells. So they basically help in uh, identifying and recognition of specific proteins present in cancer cells. They actually help the T cells in actually identifying the sites where they need to bind themselves. The working of CAR T cell therapy. So first we need to remove the person's blood, and from that blood we need to extract the patient's T cells. These T cells are then taken to CAR T cell labs, uh, so where we could make CAR T cells in the lab, which is the insertion of the gene CAR, insert gene for CAR in the T cells, uh, which would lead to chimeric antigen receptor CAR. So therefore, the CAR T cell. Then we culture it. We grow millions and millions of CAR T cells and then infuse all these cells into the patient. These infused CAR T cells can now attack the cancer cells because these CAR T cells bind to the cancer cells and kill them. So why we were using the CAR gene, because it was helping the T cells in order to recognize and attach to a specific protein present in the cancer cells and thereby attack and kill them. This therapy is considered as living drug. Benefits of the CAR T cell therapy: can treat cancer for an extended period with the potential to cure specific cancers completely. Short treatment time is needed and more rapid recovery. The challenges for CAR T cell therapy: for one cancer won't work for another type of cancer and can have negative effects on the nervous systems, risk of infection, etc. Stem cell. For the first time, a type 1 diabetic woman was treated by using cells derived from her own body after a reprogrammed stem cell transplant. So it was a reprogrammed stem cell transplant used in a type 1 diabetic women, where she was treated by using cells derived from her own body. So these cells were basically reprogrammed, and this was reprogrammed stem cell transplant. Allogenic stem cell transplantation was performed. It uses stem cells from someone other than the patient. So allogenic is basically using the stem cells from someone other than the patient. Allogenic stem cell transplantation is a kind of stem cell therapy. In type 1 diabetes, the pancreas does not make insulin, whereas in type 2 diabetes, there is insulin resistance. So in type 1 diabetes, the pancreas does not make insulin because the body's immune system attacks the islet cells in the pancreas that make the insulins. Whereas in type 2 diabetes, the pancreas makes less insulin than used to. So in the type 1 diabetes, the insulin is not produced, whereas in type 2 diabetes, pancreas make less insulin than used to. About stem cells, a cell with a unique ability to develop into specialized cell types in the body. So they can basically develop into other cell types. Two unique properties can divide over and over again to produce new cells and replace specialized cells that are damaged or lost. As they divide, they can change into other types of cells that make up the body. The major sources are embryos and adult tissues, adult stem cells. So, types of stem cells on the basis of the capacity to give different types of cells. One is the totipotent, next is pluripotent, multipotent, and unipotent. So, these are basically the four major categories. The totipotent can make all three embryonic germ layers, example the zygote. So, zygote can make all the organs. The next is the pluripotent, can make multiple lineages from all three embryonic germ layers. So these are embryonic stem cells. These can basically make multiple lineages from all three embryonic germ layers. Next is the multipotent, can make at least two different lineages, usually from the same embryo germ layer. Example, the hematopoietic stem. Next, the unipotent, can produce only one type of cell but have the property of self-renewal, which is the skin cell. So there's also a note that induced pluripotent stem cells (iP cells) are made in the lab from adult cells. So four categories are the totipotent, which can make all three types of embryonic germ layers, which for example the zygote. The next is the multipotent, which can make multiple lineages from all three embryonic germ layers. For example, the embryonic stem cells. The next is the this was pluripotent, which could make multiple lineages from all three embryonic germ layers. The next is the multipotent, which could make at least two different lineages from the same embryonic germ layer. And the next is the unipotent, which could produce only one cell type. Next is the organ on chip technology, OOC. Organ on chip technology could boost Bio3, biotechnology for economy, environment, and employment policy goal to personalized medicine for the customization of medicine. Organ on chip technology. Organ on chip technology refers to microscale system used for mimicking the human body environment. One of the human relevant 3D culture models, also known as New Approach Methods (NEMS), NAMS, 3D culture system allows researchers to recreate human organs and diseases in one dish, control the movement and behavior of materials and cells by using channels, chambers, membranes, etc. So, organ on chip technology is basically a micro scale system which is used for mimicking the human body environment. And one of the human relevant 3D culture models is the new approach method NAMS. Other new approach methods, organoids, created by growing cells from a spheroids, lead to mimic tumor behavior more effectively than regular 2D cell cultures. And the next is bioprinting. So these are the new approach methods. Bioprinting basically uses 3D printing to create living tissues and organs. So how does organ on chip technology actually work? Cells are placed on a chip and allowed to grow in 3D structures with the help of a polymer that resembles real tissue in the human body. Uses tiny fluid channels that simulate blood flow, oxygen delivery, nutrient transfer, etc., to create miniature models of biological organs, lung, heart, etc., on a chip sized device.